Our 122nd Science Cafe.
100%.
And when you talk, can you use the microphone?
If you want me to, yeah.
Paul Adams, I'm
Thank you.
And Director of our Science Math Education Institute, which sponsors this public presentation about 9 months where we have topics of interest in the science area. And I can say for those that were here for last month, yes, the satellite did not get off orbit, and we've now successfully changed the orbit of the celestial body. That's not what we're here tonight. We're bringing it back to Earth, deep into Earth, and deep into time. And very glad to have Dr. Laura Wilson here. And you see for a Hayes person, Hayes here at the Sternberg Museum, Chief Curator of Paleontology, Professor of Geosciences. And I will say that her talk, we're fortunate, she just came off her national tour and is stopping here. She went to Detroit this week and it was a similar talk, so she's just back for a national tour with that. But I'll say I saw an article about her work with this title on National Public Radio, and I said, well, this is pretty interesting. We ought to have somebody who's of national recognition here come and talk to us about what's in our very own backyard. So with that, let's give a warm welcome to Dr. Wilson.
It smells like garlic up here. It's amazing. Well, thank you for having me, for inviting me. Last time I gave a science cafe, it was the last one before we went into pandemic mode in 2020. So fingers crossed, nothing happens after this one. But I'm thrilled to talk a little bit about some of the resources that we have in our own backyard, and then kind of some of my research that I've done studying the animals that lived here 80 million years ago. Pop up somewhere. So if you're not familiar with what the world looked like in the late Cretaceous or 75 to 80 million years ago, this is it. One of the most notable things, you can kind of see where all of the continents are. They look somewhat familiar, even if they're a little crowded together, but sea level was really high. So places like Kansas and all of Central Europe and most of Australia were actually flooded with these really high sea levels, which made really unique ecosystems or really unique environments where you have very shallow marine ecosystems because they're sitting on top of continents, so very different from our deep ocean basins that we have today. If we focus in on, I don't know why Zoom keeps trying to open. If you focus in on what's going on in North America specifically, we have this seaway that is going from the Arctic Ocean down to the Gulf of Mexico and really covering, I mean, it kind of got high sea levels and low sea levels, but is covering everywhere from the drainage coming out of the Appalachian Mountains to the east. The Rockies didn't exist in this point, so really you're looking at the western shore of this being in the Utah area where you're starting to form all of these mountains of California, the Sierra Nevadas, and places like this. If we look at where we are in Kansas today, we are right about here. So we can see we're somewhere near the middle of the seaway. And we're looking at a couple of hundred kilometers, couple hundred miles offshore. So really these unique environments that we don't have any sort of analogy to today. The closest that you're going to get is the Mediterranean Sea, where we do have this shallow marine ecosystem that's covering continental landmass. But it's also very different. You just have like the Strait of Gibraltar to get in and out of the Mediterranean versus this is this very free-flowing seaway from north to south. And if we look at these ecosystems, there's some things that look a little bit familiar. We have some of these common characters that we might see in our ocean environments today, but some things are also very, very different. One of the most important differences is that we don't have any large marine mammals. If we think about some of our apex predators, our top of the food chain in our ecosystems today, we might think about our toothed whales like orcas or dolphins. We might think about our pinnipeds, so our seals and sea lions and walruses. Large marine mammals hadn't evolved yet. Instead, we have large marine reptiles. So we have reptiles that are filling these top predator roles in the ecosystem, but we still have things living along the seafloor. We still have bony fish, sharks, birds, turtles, things like that. So we have these very unique ecosystems. And if I have to talk about kind of what I do in terms of my research, I typically have a tendency to define myself as a paleoecologist rather than an actual paleontologist, because I'm very interested in animal-animal and in animal-environment interaction. So how these animals were living their lives, how it was different or similar to animals that do today. When it comes to looking at these late Cretaceous ecosystems, I do have a tendency to use this term Hells Aquarium, because we have a big ecosystem that is full of very big-bodied and big-toothed things. We have a tendency to maybe have a little bit more harmonious, look at all of these wonderful animals living together type of painting of ecosystems where it's a lot of animals eating other animals. And so I did not come up with the term Hells Aquarium, unfortunately, but I guess I am giving it a rebirth since it's the favorite description I've ever read of this ecosystem to really emphasize the number of large-bodied, large-toothed animals that we have living in these times. And because the seaway was covering Kansas 80 million years ago, actually 66 to 100 million years ago, we have a lot of these large-bodied, large-toothed animals living in Kansas at this time, so buried within our sediments. So these are just a sample of some of these Hells Aquarium predators that we have, and these are all actually all specimens from the Sternberg Museum, just kind of showcasing the abundance of these big predators. And we have everything from mosasaurs. So as I mentioned, we didn't have large marine mammals. Rather, we had large marine predators. So this top one, this Tylosaurus that we have at the top image, if you've been to the Sternberg Museum recently or since 1999, this is the one that takes up the entire back wall of our fossil gallery, and it's about 30 feet long, with this big honking head with big teeth in it, sitting as one of our top predators that we have in the seaway. And all of these are different types of mosasaurs, and mosasaurs are we've been very fortunate to have many mosasaurs with gut contents, so actually remnants of their last meals within their stomachs, and we see everything from bony fish to sharks to turtles to seabirds to other mosasaurs. So we have a very good idea that these were just big vacuum cleaners that would pretty much eat anything that would fit in their mouth. We have other types of marine predators. So tylosaurs were very interesting, or mosasaurs were interesting because they're this unique kind of love child of a monitor lizard and a snake, that they have an extra set of teeth on the top of their mouth, in addition to having all of these jaw joints that allow them to open their mouths even wider. So think of a snake eating, but with actual 3 pairs of jaws instead of 4. We have other things here like plesiosaurs. So if anybody's up to date on their Latin, did anybody take Latin or Greek? So megacephalosaurus, can you translate it? Big-headed lizard. So the name gets right to the point that you have this huge head on this big predator with big teeth. They're conical, they're pointed. They are all about kind of puncturing and ripping and gripping different prey items. If you've been to the museum, this is the big one that's hanging from the lobby as you enter the museum. We also have things that are a little bit more familiar to us today, like sharks. So this is one of my absolute favorite specimens that we have at the Sternberg Museum. And if you know one thing about sharks, it might be that they have their skeleton made out of cartilage. So what we make our nose or our ears out of is very flexible. It allows for a very powerful and fast and agile body, but it does not fossilize. So unlike our bones, which we put some calcium phosphate, some actual mineral into, cartilage pretty much typically doesn't fossilize. I'm going to play this again just because I like it so much. And nope. Sorry, my Zoom keeps trying to load for some reason. But one of the things that you can see here is you can see all of these teeth that are actually preserved as if they were still embedded in the cartilage of the jaw. But the cartilage has been dissolved. So they're preserved in this perfect 3 dimensions, even though their cartilaginous jaw structure is gone. It's just the coolest specimen. There are also scales, so the actual scales that cover the body are preserved as well. It's really, really cool. This one's from Ellis County, so right from our back door. The nickname for the Cretoxyrhina, so the technical name, I have some of these teeth up here that you can see after the talk. The nickname for this shark is the Jinsu shark, named after the Jinsu knife, which was all about slicing and dicing. So just to give you the reputation of this shark that we have in these ecosystems. And then, of course, kind of the poster child of the Sternberg Museum is Xiphactinus. So the fish within the fish. Xiphactinus here is the big fish with a smaller, although a 6 to 7 foot long fish within its belly, so that small fish is still pretty big. And so just showing again, big fish, big predator, eating other big things. These are also called bulldog fish, and I am a big University of Georgia football fan. So you can kind of see some of the similarities that we have in their jaw structures with this upturned jaw that we have, so probably big attacking predators from below. These also look very similar to modern day tarpon, so probably filling these big kind of apex bony fish roles within the ecosystem. And this is a really cool specimen as well because we actually have evidence of these ecological animal-animal interactions. So rather than just a big fish maybe happenstancely falling on a smaller fish, the smaller fish, the gillicus in its belly, is actually trapped within the rib cage. So we have a pretty good idea that this was a failed, the big fish did eat the little fish, but then the little fish somehow ended up killing the big fish. So some sort of failed or /successful predation event. So lots of big toothed things that we have filling the top roles of the ecosystem. And then one of these Mesozoic Age of Dinosaur characters that's missing are the actual dinosaurs. So we don't actually get many dinosaurs or dinosaur fossils within the sediments that we have out here in western Kansas. With the exception of birds, because birds are dinosaurs, and that's an entirely different subject for an entirely different talk. But with the exception of birds, dinosaurs lived on land. And no matter where you stand on the whole Spinosaurus was aquatic or debate, which is a very big debate in paleontology right now, even Spinosaurus would be in large river systems, not out in the middle of oceans. So dinosaurs were on land, and then we had other types of marine reptiles in the ocean. So you have a very tendency to die and get preserved in places where you don't live. The very few fossils that we find out here are through this very special process, which is one of my favorites, called bloat and float. So people who may have grown up in rural areas may be familiar with what dead cows look like after they die. They have a tendency to fill with gas. And this is just a byproduct of the decay process. The bacteria that consumes soft tissue releases gas. So it kind of respires and breathes the same way that we do, breathes in most case bacteria, oxygen, and then breathes out CO2 and things like that. So that product of respiration then inflates the body. So I had a student, one of my first students, who very lovingly called these bloat and float animals meat balloons. And this is exactly what they did. So things like Niobrarasaurus was this big armored dinosaur. They're kind of built like tanks, that they have all of these thick scales on their backs and bones on their backs. So if you kind of turn them upside down, it's kind of like a boat, just floating out in sea. So the idea is that they would die on the shoreline, probably over actually on the Utah side. And then they would start to decay, and then tide would come in and pick them up, and tide would go out and carry them out to sea, and then they could float for hundreds of miles or kilometers out to sea, to the point where they would kind of be like a whale fall, and go through the stage of floating out to sea, to where eventually they would become waterlogged, or other animals would start to eat them and kind of pop them, and then they would sink to the bottom. So we do see this with dead whales today. Between the blubber and the bloat and float, they float for a while, and actually this big feast for aquatic animals, before they then float down to the bottom to get preserved. So this is not a Niobrarasaurus dinosaur, but kind of an example of the model that we might see, where we have this dinosaur floating out to sea and then being a feast for animals and eventually falling to the bottom. And that's what we get here in Kansas. So one of my favorite stories of dispersal of skeletons, but one of the rare occasions where we actually do get dinosaur fossils preserved in our western Kansas sediments. At this point, I was trying to figure out how to transition to starting to talk about some of my research on these animals. And I was like, okay, well, I want to talk about those animals that aren't the top predators. So the idea that not everything that lives in the seaway is big and toothy. And then I realized that this is just really, in fact, a lie, that pretty much everything is big and toothy. So I studied these birds on the left, so Hesperornis, and these might not be apex predators, but they're like 6-foot-long birds, and from their feet to the tip of their beak, and they still had teeth, so very different from all of our birds today, which are completely toothless, except for a couple of a mutant chicken every once in a while. But for the most part, we have toothless. These are toothed big birds, so lie. And then I also studied these turtles, Protostega, which you can see are like 9 feet long. So they might be lacking teeth, but they're still huge. So trying to think about kind of how to rebrand what I'm studying, it's that not everything isn't necessarily big and toothy, but it's really about how to survive in this Hells Aquarium when you're not a top predator. And that's really what I'm going to be talking about with my research, is some of these things that weren't necessarily going around eating everything that could fit in their mouths, what they did to survive when it seems like all of these animals in these ecosystems are big and toothy and out to eat you. But before I can talk about some of the specifics of these animals that I've researched, I want to talk about how I undertake my research. So I use a tool called osteohistology. So going back to our Latin and Greek roots, osteo is bone, histo is tissue, and ology is the study of. Sorry, I took a lot of Latin in middle school and college, so I like to break things down into roots. So I study bone tissue. And this is really interesting because if you study bone tissue, you can really understand how animals grew throughout their life, which is really important for putting together the evolutionary history or the function of an organism or how it lived in its environment. So what I do is I take bones and then I actually cut them up. So this is often termed destructive analysis. I prefer to call it consumptive analysis, because even though I am permanently altering these bones, I'm actually getting more information out by cutting them up than you could ever get just by looking at the surface. So I take these bones and I put them to different size saw, everything from these small saws like you see here with a diamond encrusted blade to like the big tile saw for if you're renovating a bathroom or a kitchen that you use to cut porcelain tiles. And then I actually put it on these grinding wheels and grind it down really, really thin to the point that I can actually shine light through it and look at the actual bone tissue that is deposited while that animal is alive. So if I take an example of this, I can take, this is the upper arm bone, the humerus of a chinstrap penguin, which I have a cute little illustration of there. So a living modern animal, and you can notice it's kind of flattened because aquatic birds have a tendency to evolve their wings into paddles, because even though penguins don't fly in the air, they actually fly underwater. It's the exact same flight stroke. So they have these elongate bones, but we can go in and look at more detail to see some of the microstructures. So if we take one little area, we can zoom in and see all of these details within the bone itself. These big holes that we see that are oriented in a bunch of different ways, these are the vascular canals. These are areas where the blood vessels are bringing blood and oxygen and nutrients to the bone. And we have studies of living animals, which are dead by the time we do this research on them, to actually be able to trace the growth rate, so how quickly these animals are growing based on the number and the size and the orientation of these vascular canals. You can imagine that the more blood and nutrients that you have coming into the bone, the faster that it's going to grow. And we can actually study this in living animals where we can say that they're growing this large or this number of pounds within one day. We have other features that we can see as well. So all of these little black specks that kind of look like poppy seeds on the top of a bagel, these have the very scientifically intimidating name osteocyte lacunae. And this is just a fancy term for kind of like a little apartment where the cells that lay down new bone live. So each one of these black dots represents a bone growing cell. And we can look at the number and the shape and the size of these cells, again, to look at growth rates. So all kind of different evidence that we can test with living animals and then take back into the fossil record. Because the really cool thing is that all of these features actually fossilize. And I can show you this. So my next set of slides is actually looking at Pteranodon pterosaurs. So pterosaurs that we find in the seaway. And this is looking at the exact same bone under different types of lighting. So one of the things that you see moving from the left, which is plain light, so what this light would look like through sunlight or an incandescent bulb, all the way to polarized light, which actually focuses light in a single plane. And then the one on the right where I take this light and then just pass it through a filter that moves everything to the magenta end of the color spectrum, which just makes it look really pretty in purple and magenta. But what this is showing is the actual protein of the bone. So one thing to know about bone is that it's kind of like reinforced concrete. If you've ever seen reinforced concrete get poured, you have concrete into this matrix of rebar. And bone is built the same way, is that we have this crystalline structure, this calcium phosphate, and that's the actual mineral, and that gives our bone strength. And then within that, we have protein. We have the collagen, or the protein collagen. So the same thing that we build our hair out of and our fingernails, things like that. And that gives our bones flexibility. So if you fall down, unless you have problems with too much calcium or not enough protein, you have a little bit of bend in your bone, that they're rigid enough to give us strength within our skeleton, but we can fall down and not necessarily break ourselves all the time. If you look at little kids, they're very bendy because they have so much protein and they don't have as much of that, that mineral in them, that hard mineral. So this is actually showing that even in fossils, the protein, that soft tissue, gets preserved. And if you think about your day, when you're going around really, really quickly and you don't have a lot of time, you're really unorganized, right? Everything's all helter-skelter. Versus if you are very calm and relaxed, you have a time to get all of your ducks in a row, and you can be very well organized. Bone is the exact same way when it grows. That when bone is growing really, really quickly, like when we're little kids, we get this kind of crazy helter-skelter pattern, and there's actually kind of no pattern that we can see. When bone growth slows down, it becomes very organized. So we see things like we see in the second and third or the middle and right photo where you get these banding patterns. And this is showing us that bone growth is starting to slow down. So we can look at all of these bone growth patterns to look at relative age, or relative growth rates. What animals are growing quickly, what animals are growing slowly. And this can relate to things like the evolutionary history of an organism, so what type of animal it is, how old it is, whether it's young, whether it's old, how it's using that bone, or any sort of environmental pressures that may be going on. Another thing that we can do with this technique is looking at absolute growth. So the only way that we can actually say how old an animal was when it died is to use this technique of osteohistology. So I showcased this image as part of an exhibit I put together for the Sternberg Museum, actually showcasing some of my research images as art. So you can go to the museum and see that today or tomorrow. But I showcased this at one of our art walks. And the most common comment I got about this, because I forgot all of my little label cards saying what anything was that it looked like cork. And the same way that and cork is tree. Is wood. And so the same thing that we can kind of cut open a tree and count rings, we can actually do the same thing in a lot of bones. So all of these lines that look like tree rings are actually growth lines, so the same sort of thing. The only difference is that this is a turtle. So this is a turtle leg bone versus a tree. And all of these concentric rings that we see going around the bone are growth lines. And we have these in our bones as we get older. We just do really weird things like modify our skeleton and modify our environment, and that's a whole other story. But you can see these lines. So this animal, the sea turtle, was actually 4 years old when it died. So we can actually go back in and say, absolutely, this animal was 4. And this is something that we can't do without this research, this style of research. But all in all, we can put things together and really look at relative growth ages or growth rates. So what animals were growing quickly, what animals were growing slowly. So I have the upper arm bone of 2 different types of birds, where a penguin is growing their bones very quickly, and an emu is growing their bones relatively slowly. And this is all related to penguins use their arm bones to swim, and emus pretty much don't use their arm bones. So it's not necessarily to grow for them to put as much energy into it to grow it as quickly. So this is a little bit about the technique that I use to get back to what's going on with some of these non-apex predators and their survival strategies within this seaway. So the first thing I'm going to talk about is Hesperornis. So these birds, I love these birds, they're so goofy, and they have these really unique skeletons that tell us a lot about how they were living their lives. And they have a very unique suite of characters. So everything from what we see in their forelimbs. So if you're somebody who prefers the white meat of a turkey or a chicken, what you're actually eating is the flight muscle. So it's those big muscles that attach to the breastbone that are used for flight. The big breastbone, which we call the sternum, has this big keel on it, like the keel of the ship, that provides all of this surface area for muscle attachment. So if you have a big keel on your sternum, you're going to have big flight muscles, which you're going to need for flying. If you are lacking any sort of keeled sternum, you're not going to have big flight muscles, so you're probably not going to be flying. Other things that they have is they have these tiny little arms. They have one bone. They have their upper arm bone. They're completely lacking their forearm, their wrist, their fingers. So they really just have these little toothpicks that kind of sit out of their shoulders, stick out of their shoulders, so they're not really going to be using their arms for flying. If we look at what's going on with their legs, they have these, their femur, their upper leg bones, which really stick out of their hips at a 90-degree angle. And versus sitting underneath them like we have, they stick straight out. They have these big, huge hips, all for these big, powerful muscle attachments. They have very long shin bones, so their lower legs are elongate, and then they have these big, huge feet. So they have tiny arms, they don't have flight muscles, and then they have these big, powerful legs, telling us that they are going to be using their legs to swim underwater to pursue prey. I have an example, I'll show it after the talk. I have an example of the leg and kind of what it looks like when all of these bones get put together. So we can tell unique things about how they survived. And the key thing here is that they are flightless. So they are not going to be flying from place to place. So when I was studying these, I was really interested in their distribution. So all of the dots on this map shows where all of fossils from these types of birds have been found all over North America, all the way down from Arkansas in the south up to Ellesmere Island and the North Slope of Alaska to the north. And the main question that I was really interested in is, were these birds migrating? Did they just maybe have a really large home range and were found all over at all times of the year? Or were they living in the south in the summer, living in the south in the winter and then migrating north in the summer? And we can look at what this would mean environmentally, so we can put some temperatures on this with really these southern latitudes, kind of Gulf of Mexico area, very similar in temperatures to what we find today. But then we go up into the Arctic, and it was much, much warmer in the Cretaceous versus today. There are no polar ice caps, just one of the reasons why sea level was so high. And we're looking at, maybe 10 degrees Celsius. Can you translate that to Fahrenheit? What's 10 degrees Celsius to Fahrenheit? 50 degrees warmer? 50 Fahrenheit. So probably about 40 degrees warmer Fahrenheit than what we found today. So where we might be around like 2 to -2 mean annual sea surface temperatures in the Arctic today, we're looking at 10 to 15. So very, very significantly warmer. One of the things that is the same is that these high latitude Arctic ecosystems would still undergo 24 hours of darkness during the winter. So even if it's much warmer, you're still cutting out sunlight for 3 months, which means that you're cutting off the base of the food web. The base of the food web is going to be plants and algae and all of these organisms that photosynthesize, so take sunlight to produce energy. If you don't have sunlight, they can't produce energy. So somehow anything that would overwinter in these high latitudes, it might not be as cold, but there'd still probably be a period of starvation or something like that. So I took bones from both the Canadian Arctic, so from Devon Island in the Canadian Arctic, as well as from Kansas. And I cut them open, asking the question of how these birds grew at both mid-latitudes, so in Kansas, as well as high latitudes in the Arctic. And the main thing that I found when I cut these bones from different ecosystems was that they looked pretty much exactly the same, that they had rapid but consistent growth all the way through them, so pretty much from hatchling to an adult. They grew the same. And I didn't find any differences between Kansas bones and Arctic bones. But then I reached the problem that I didn't really know what this meant. So one of the things that I do in all my research is I find a modern analog and I see what modern ecosystems are doing or what's happening in modern ecosystems. And for this, I specifically chose penguins. So Hesperornis, these fossil birds, are not closely related to penguins, but they've actually fill very similar niches within ecosystems. They have a wide range of body sizes, and they're found at diverse latitudes. They're kind of found all over the southern hemisphere, and they have a variety of behaviors. I took 3 species of penguins specifically. They're all in the same genus. I took the Adelie, the chinstrap, and the Gentoo penguin. So they're all very closely related to each other evolutionarily. So there's not going to be a whole lot of difference related to their evolution. All of the specimens that I used came from the Antarctic Peninsula, from the same localities, and so they're all found and lived in the same area. They do have a variety of body sizes, with the Gentoo having a larger body size than the Adelie or chinstrap. But one of the most noticeable differences is in their behavior. Where the Adelies and the chinstraps are migratory, but the Gentoos are non-migratory, which means they pretty much spend their entire lives around where they hatch. And then they go back and lay their eggs and reproduce at those same sites. Versus the chinstraps and the Adelies might migrate anywhere from 100 or 1,000 to 3,000 kilometers within just the winter season trying to find food. So this was a great little sample set that I found where I could specifically test the effects of behavior on bone growth, specifically migratory behavior. And what I found, I'm not expecting you to do kind of any sort of in-depth analysis on this, but my Adelies and the chinstraps grew very, very similar. Their bones look nearly identical, where the Gentoos' bone growth is different. And more specifically, it's different in a way that indicated that Gentoos grow faster. So they're growing larger at a faster rate than the other 2 species. The main reason for this, as I interpret it, is because they're non-migratory, because they have to overwinter in harsh conditions, and so they have to grow large enough to be able to undergo a period of essentially starvation. So they have to gain enough weight and gain enough body size by the time winter comes so that they can then starve off some of that body weight without dying to then make it to the spring season. And we see this in a couple of other species of penguins as well. So then I took these results back to my Hesperornis birds and determined that the lack of differences that I see in my Kansas and my Arctic Hesperornis is the same sort of thing that I see in my migratory birds, is that I kind of put 2 points together, that migration does not leave a mark on bones, which I found from looking at my penguins. But then overwintering does. So the fact that my fossils look the same, I'm interpreting as that they were migratory, and I don't have any evidence of overwintering. So if I put this back into my survival strategies in Hells Aquarium, my survival strategy number 1 for these birds is get the heck out of Dodge. That you move ecosystems, and this helps you go maybe north to reproduce, where there's less chance of predation upon your young, like we have a lot of our migratory birds do today. And then you overwinter in the south, where there's going to be higher food abundance because you're not going through a 24 hours of darkness period. So then this takes me to my turtles. So specifically looking at Protostega. So Protostega is the second largest sea turtle known, or second largest any turtle known of all time, second only to Archelon, which is his like direct descendant. So a lot of evidence points to Protostega evolved into Archelon, and it just kept getting bigger and bigger. But you can see it compared to the leatherback sea turtle, which is our largest living sea turtle. So they're about maybe up to 6 feet long, and Protostega is probably about 9 feet long. So a really, really large sea turtle. Their distribution compared to what I just talked about with Hesperornis is a little bit different. They don't have this broad distribution necessarily that we see. And what I was looking at with Protostega is actually specifically only at individuals found here in western Kansas. So only those in our chalks. because I wasn't setting up a comparison between southern latitudes and northern latitudes, I actually want to know how these sea turtles grew throughout their lifespan. And I did this by putting together a series or a growth series where I got small individuals, hoping that they were younger, and large individuals, hoping that they were older, so I could see how they aged. And if you're not up on your metrics, something like this big one is about half of the length of this femur is pretty much the length of, or sorry, humerus is pretty much the length of mine. So that second to the last one, the CM1421, this is its size. And the one modeled in that picture is even larger than this one. So these are very large sea turtles that's just one bone of its flipper. So the first one I looked at was one of these smaller ones. So the image you see on the right is the complete cross section, and then the focus is kind of some of the highlights under magnification. If you look at the one on the left, it looks like a sponge. So this is actually called spongy bone because it has these big holes in it, which is a factor of it being very large and needing to stay buoyant in water, but is also a factor of very large growth. And I can actually go in under microscope and count these growth rings. So this animal was 4 years old when it died. So showing me that it's actually laying down bone really, really quickly. So overall bone growth is fast. I can then compare it to my largest sample within the data set. And I see very similar things, that I still see this spongy bone texture. And then I can go in and count rings. This isn't the complete cross section, but this one has 8 rings in it. So essentially a turtle this big, a little bit bigger than this, was 8 years old when it died. So I can compare these two and look that between 4 years and 8 years, it essentially doubled in size. And that's really, really fast bone growth for a turtle. I mean, this is fast bone growth for a mammal, but especially for a turtle, which is cold-blooded and supposed to have slow bone growth rates. And I ran into some of the same issues that I did for my Hesperornis with what does this mean? How can I make interpretations just from knowing bone growth? So again, I went to modern animals. And this analogy was a little bit more obvious because sea turtles are alive today. And I went to the modern sea turtle record. So one of the interesting things about sea turtles is that they are still alive and they're actually very well studied. And this is because you can't go out and kill sea turtles. They are all protected, they're all threatened, and many species are critically endangered. So we try not to kill those that we know are already going extinct. But one of the things that we can do is study their already dead bodies. And this is what a lot of turtle conservationists do, is they take already dead stranded sea turtles, they cut open their bones, to understand how old they were when they died so they can do population modeling. And this is how they do pretty much all of their sea turtle conservation, is by cutting up dead animals and looking at how they're growing. And if I look at the modern sea turtle histology literature, you see that there are 2 different types of bone growth within modern sea turtle. On the right here, I have the leatherback, so these big sea turtles that are growing very, very quickly. So you can see that pink spongy bone. It's only pink because it's dyed pink. It's not pink in real life. But you see this spongy bone that's growing very, very quickly. All the other types of sea turtles, so your greens and your ridleys and your flatbacks and your loggerheads, they're in this completely different evolutionary lineage that grow much, much slower. And so they have this kind of dense bone and a very different growth pattern than what we see within our leatherbacks. So if I compare my protostegas, my fossil bones, to modern sea turtles, I see that my fossils are growing much more similar to leatherbacks than they are to any other type of sea turtles. And this is really interesting because not only do leatherbacks grow really quickly compared to other sea turtles, but they have very interesting life histories. And when I say life histories, I mean when they're hitting very important milestones within their lives. So how quickly they're growing, what their metabolism is doing, when they hit full body size, full size, and when they hit sexual maturity, so when they can start reproducing. And leatherbacks are very unique in that they have a large body size, that they grow to very quickly compared to other, even much smaller sea turtles. They live their entire lives at sea. They migrate into Arctic waters, which is really weird because we don't think about reptiles going into cold areas. They can also dive to like 3,000 or 4,000 feet deep, so really, really deep, cold waters, which is unique. And then one of the coolest things about them is that they actually hold a body temperature higher than their surroundings. So if we think about reptiles, snakes, lizards, and if anybody's ever had a pet reptile, you know that you have to keep it under a heat lamp, essentially because they absorb heat from their environment to get enough energy to go about their daily lives. Animals or reptiles that live in colder areas have a tendency to like hibernate or go through a torpor and kind of shut down their systems during cold times. But these leatherbacks actually can survive in these much colder environments because they can hold a body temperature. So something that's very, very unique to leatherbacks. So I made a lot of, based on these comparisons, because the protostega, my fossil sea turtles, are growing their bones in very similar ways, and bone growth relates to metabolic rates, that I'm making a lot of comparisons that they're also probably living their lives in very similar ways in terms of growing fast to a very large body size, reaching sexual maturity very quickly, which is a wonderful survival strategy evolutionarily. The faster you can reproduce, the more young that you can have, and the higher chances of survival of your species. And putting this together, looking at my growth rates, is that you have a 9-foot-long sea turtle that could possibly be reaching full body size and the ability to reproduce within 10 years, which is really, really fast for pretty much any animal, much less a turtle. So this brings me to my survival strategy number 2 within Hells Aquarium is grow up fast. And this is, I think, how one of the best survival strategies against getting eaten is body size. It's very hard to eat large things. So putting it together in terms of Hells Aquarium, that if you're not an apex predator, you better grow up fast or get the heck out of Dodge. So thank you all very much for your attention. I'll be happy to answer any questions you may have. I also have some fossils and casts up here for you to look at for some of the animals I've talked about. And thanks so much.